Hello, my name is Ryan Hesterman, and I'm a junior neuroscience major.
Hi, my name is Shannon Rose. I am a senior neuroscience major.
Hi, my name is Sophia Pinziucci, and I'm a junior biology major.
With the mentorship of Dr. Kristen Ackerman, director of the neuroscience department at High Point University, we have developed a paper that explains how Playdoh can serve as a 3D model depicting cellular movements and tissue remodeling during gastrulation, early organogenesis, and embryonic folding.
In addition to the paper, this video will provide a visual resource to demonstrate how these processes can be modeled through the use of Playdoh.
The following video will provide a step-by-step instructions on how to model this in the classroom.
We will start with two balls of cells known as the epiblast and the hypoblast. The epiblast is represented by the blue Playdoh, and the hypoblast is represented by the yellow Playdoh. These balls of cells will begin to flatten to form the bilaminar disc, which is going to begin to develop as the blastocyst implants into the uterine wall between day 8 and day 9 of development. Students will be instructed to use a rolling pin to flatten out these balls of Playdoh. Students will continue to use the rolling pin until a nice flat surface is formed. Students will then use a straw to represent cells by poking the tops of these, and will continue to do this until cells are formed on the entire tops of the layers. Students will then use a ruler to measure out 5 inches by 3 inches for each layer. The epiblast is placed dorsal to the hypoblast. From here, the first developmental structure that will form is the primitive streak, which forms at the caudal end of the epiblast. This establishes the dorsal ventral axes, or the top and bottom of the embryo. The primitive streak splits through the epiblast, but does not go into the hypoblast. The epiblast cells will then delaminate from the lateral edges and be moved toward the primitive streak. In order for the epiblast cells to fit through the primitive streak, they must flatten into a bottleneck shape. Once the epiblast cells have passed through the primitive streak, they integrate into the hypoblast, or the yellow layer, and form a new layer known as the endoderm. Students will continue to show this migration of the blue Playdoh going down into the yellow Playdoh to form the endoderm. Students will also be asked to use yellow strips to show how the blue Playdoh will begin to turn into the endoderm layer. Epiblast cells will continue to migrate through the primitive streak until the space between the hypoblast and epiblast is filled, and will create the middle layer of the trilaminar disc, known as the mesoderm, and is shown with the red Playdoh. Students will also use strips to show the beginning of this structure. Then students will use a fully formed red layer to show the fully formed mesoderm structure. The remaining epiblast cells become ectoderm. Now the trilaminar gastrula is fully formed, with the endoderm on the bottom shown with the yellow layer, the mesoderm in the middle shown with the red layer, and the ectoderm on the top shown with the blue layer. After the formation of the trilaminar gastrula, the next structure to form is the notochord. The notochord is a rod-like structure that forms in the cranial end of the embryo, opposite of the primitive streak. The ectoderm that is touching the notochord will become neural tissue, and the rest of the ectoderm will become hair, skin, and nails. The formation of the nervous system then occurs by the ectoderm beginning to expand to form the neural plate. Students will represent this by adding blue Playdoh attached to the ectoderm structure. The ectoderm that is touching the notochord is pulled toward the notochord to form the neural groove. This is represented by pressing a pen down into the ectoderm that sits above the notochord, forming the neural groove, neural folds, and lateral edges. Pressure is then exerted on the lateral edges to form a neural tube. First, the middle closes, then the caudal uncloses, and finally the cranial uncloses. The neural tube is now fully formed. While the neural tube is forming, the perimeter streak will regress slightly toward the caudal end. Finally, once the neural tube forms, the cranial end will expand and proliferate to form the brain. While the nervous system is forming, the cardiac formation occurs at the same time. This happens by cells from the mesoderm migrating through the perimeter streak. These cells will migrate around the forming nervous system and will create a cardiogenic crest around the brain that has already formed. This occurs around day 15 of development. These cells will loop into the heart by day 28. The heart is anterior or cranial to the brain, and folding will allow the heart to move into the correct position within the chest cavity. Folding then begins at the cranial end due to the size and weight of the developing brain. This will allow the heart to be in the correct anatomical position. The tail will then fold, followed by the lateral edges, wrapping around to close off the ectoderm. This model shows the fully formed ectoderm around all internal structures. This example represents the mesoderm outside of the ectoderm. This example represents the brain that is formed outside of the ectoderm.